thank you very much to Margaret for that introduction, and thank you to the Alumni Ambassadors for inviting me to start off this speaker series. I'm really excited to be here. It's really great to be back on campus. I think I haven't been back since 2018, so I'm really glad to have the chance to be back. And thank you to everyone who's come out today to listen. So my presentation is called Mycobacteriophage to Mycobacteria, and this is in reference to the research that I did here at WKU with Dr. King. I researched on mycobacteriophage, which are viruses that infect a certain type of bacteria. And now in graduate school, I've been studying the bacteria rather than the viruses that infect them. So this is sort of a reflection of the journey that I've kind of taken from WKU to where I am now. And as Margaret said, I'm currently an MDPhD candidate. I've already have the PhD part, but I'm still working on the MD part. And I'm in New York City at the Tri-Institutional MDPhD program. So I won't read all the names of the schools to you guys again, but we're called Tri-Institutional because there's 3 schools in our program. So today I really want to talk to you guys a little bit about my experiences at the Honors College and at WKU. And then I'll use those to kind of show you how the experiences that I had at WKU have really helped me in the experiences that I've had after WKU. So that will be the first part of the presentation. And the second part, I just want to give you guys a little bit of an overview of the research that I've been doing for my thesis and the research that my PhD was based on. So, and then there will be some time at the end. If anybody has any questions, I'd be very happy to answer any questions over either my experiences or the research part. So I realized today that I'm very old. This is what WKU looked like when I was at WKU. So you'll notice that it's basically the same, but there are a couple of differences. One major difference is that the building that we're in right now did not exist. So this is a new addition to WKU from my perspective. And so actually when I was at WKU, the Honors College was this little tiny building over here. And the Honors College was the little house, and behind it there was a trailer where the Flagship Program and OSD were lived. So Dr. Jennings remembers this. And I spent a lot of time in her office in the little trailer over there on this little street corner. So I think this building is a major improvement. I'm really glad that you guys have this lovely building to be in. But we had a lot of good times in that little small trailer also. So as Margaret mentioned, I did research in Dr. King's lab. So his lab was here, and I was happy to find out today that it's still there. So I got to take a little tour, which was very fun. And because I was a science student, I spent most of my time in these buildings over here. And you'll notice that those buildings are still there, but the names are a little different because this building has been replaced by a brand new building, which I got to take a tour of today, so that was very fun. And I also had 2 homes away from home, so I was a Gatton student and I lived in Florence Schneider Hall. And then when I became a full-time WKU student, I moved to McLean Hall, and I spent the time that I was actually in the country in McLean Hall. So I got to walk past the colonies every day on my way to my classes, and that was always a very nice walk. I missed it being in New York City. So I just wanted to give you guys, this is where I'm coming from. I graduated in 2014. This is a timeline to tell you guys about all the things that I was able to do at the Honors College. So as Margaret said, I started out as a Gatton Academy student in 2009. And I think I was in the 4th graduating class of the Gatton Academy, so that's also very different. The Florence Schneider Hall is much bigger than it was in my days. But while I was at WKU, I actually had the opportunity to join 2 new programs that had just gotten started at WKU. And the first of these was the C-Phages program. And this is the Genome Discovery and Exploration program, is what we call it at WKU. So I got to join Dr. King and Dr. Reinhardt in that program. And that was where I got my first taste of scientific research. And so basically what we did in that class, it was like a structured introduction to research. So I got to start doing some research on bacteriophage, which again are viruses that infect bacteria. And that was where I really got excited about biology and about doing research. And that was what really sparked my interest in doing an MD-PhD later on. So the other program that was just getting started at the time was the Chinese Flagship Program, which some of you might be familiar with. And I'm also very happy to see how that has expanded over time. So I was part of the very first class of students in that program. And these 2 programs really formed the cornerstone of my experience at WKU. So they have been very, very important in everything that I experienced, and everything that I experienced since then has been informed by the experiences that I had in those 2 programs. So to kind of move along on the timeline, through the Flagship Program, I had the opportunity to study abroad in China several times as a Gatton student. And the first of these was winter 2010, and then summer 2010, and then winter 2011. And because I was a science student also, I also had some opportunities to participate in research during these times. So I joined Dr. King's lab shortly after I started in the phage program. And I also was able to do some independent research in his lab during the summer of 2010, I think around the time that I spent in China. And this is where I started doing the thesis, or the research that made up my thesis when I was for my honors capstone experience. And then in the summer of 2011, I also I applied for a lot of different research, undergraduate research programs, and I was accepted to one at Princeton University, and here I had a lot of help from one of my mentors at the Gatton Academy, Dr. Derek Schrader. And so I was able to go to Princeton, and I studied yeast genetics at Princeton, so I kind of got the chance to try out some different type of research within a different environment. So that summer, I also made the decision to stay at WKU to finish my undergraduate studies, and that was really because I had such great mentors on the science front, and I also was really excited about continuing with the Flagship Program. So those were the two things that really inspired me to stay, as well as the experiences that I knew I would be able to have in the Honors College. So I stayed, and I started as a WKU student in 2011, and apparently I spent one semester taking classes, and then I decided that I wanted to kind of explore some new frontiers, even though I had already started doing that a little bit. So I actually talked with Dr. Jennings, and she let me know about this program called Teach and Learn in Korea. And we had an alumna of the Honors College who had done a similar program, and so she was able to help me with that application, along with Dr. Jennings. And I was awarded this scholarship by the Korean government to study, or not to study, but to teach English in South Korea for 6 months. So I spent spring and summer of 2012 there. And this was in a very, very small elementary school in a very, very rural area of South Korea. And I was 18 years old and all by myself, so this was a very interesting experience. I learned a lot of things. I think I gained a lot of independence during that time. And I think it was a really interesting thing to have on my resume. So I came back from that in July of 2012, and then I was ready for my capstone year for the Chinese Flagship Program. And so in August of 2012, I headed off to Nanjing, China to start my capstone year. So I was actually the first WKU student to do the capstone year as part of the Flagship Program. So I didn't really know what to expect, and I was on my own still. So I headed off to Nanjing, and I was able to make a lot of friends from other universities that participate in the Flagship Program, as well as at the university among the Chinese classmates that I had there. And so a part of the capstone program is that the first semester you study at a university, and then in the second semester you are supposed to find an internship in your field. So I was a biology, chemistry, and honors self-design studies triple major. And the third one was how I was able to package my Mandarin classes into a major, because we didn't have a Mandarin major at the time. So I wanted to try and find an internship that related to biology and chemistry. So of course I reached out to some different PIs at different researchers at institutions around China, and I asked them whether they would be willing to have me research in their lab for the spring semester, and I found one in Shanghai at the Chinese Academy of Sciences who was willing to have me come. So I spent the spring of 2013 doing research in a cancer lab at the Chinese Academy of Sciences in Shanghai. So during that time, I was also applying for summer programs for research opportunities in the United States, and that was with the help of Dr. Jennings. And so when I came back from China, I headed off to New York University, and there I did stem cell research for a summer. And then I spent a final year at WKU to finish up my coursework, and also to finish writing my thesis and defending my thesis. And so my thesis was based on the research that I had been doing in Dr. King's lab on the characterization of the bacteriophage that I isolated in the phage program. And so I wrote that thesis, defended it, and it was a really good way to kind of wrap up my experiences at WKU. And so before I moved on to my next step, I also spent a lot of time in the office of Dr. Jennings and other people in the Office of Scholar Development, working on a bunch of scholarship applications. So I was able to apply for a number of different national scholarships with the help of Dr. Jennings. And I was named a finalist for these 4 scholarships, and then I actually was able to get these 3. So these 2 are ones that are for science students who want to do PhD research in the sciences, and this was a critical language scholarship to study Korean. So I had to decline these because they were not compatible with my next step, and my next step was to join an MD-PhD program in New York City and continue researching and kind of developing my scientific skills. So I learned a lot of things as an MHE student, and I want to kind of summarize those a little bit here and then show you how they have helped me get to where I am and how they've helped me in the position that I am now. So one thing that I learned, of course, was cross-cultural communication. So I learned this through my study abroad experiences. And this has been really helpful, I think, in where I am now. A lot of people in my lab come from different countries, and it's been really helpful to know how to understand their different backgrounds and to be able to communicate with a lot of different kinds of people. I learned the importance of mentorship. So I mentioned a lot of the mentors that I had at WKU and in the Honors College. And these people were very, very important in helping me to get admitted to the program that I was ultimately admitted to, and also in teaching me what to look for in a mentor. So I knew that I had had such great mentors at WKU. I was able to look for those same qualities in mentors that I chose in my graduate studies. So I also learned long-term and short-term planning. And one of the very first things that I did after I decided to stay at WKU was to meet with Dr. Jennings and make a long-term scholarship plan for all the scholarships I was going to apply for. So we mapped out when I would apply for each one. And then, of course, each individual scholarship had a short-term plan where you map out when you're going to complete each part of the scholarship. And so those skills have really come in handy in the lab. So on my research that I've been doing as a graduate student, you really are thrown into the lab and you don't really get a concrete plan of what you should do every day. So you have to kind of plan things out yourself, and of course you have to have some short-term planning for different experiments and long-term planning to know how things are going to fit together at the end. So those have been really helpful skills to have as well. Going along with that, time management is really important. I kind of learned that when I was trying to work on my thesis at the end, juggling classes and interviews and writing that. So I was able to develop those skills, and those are, of course, have been very important in where I am now. How to write applications. As you might guess, I wrote a lot of different scholarship applications, and I think through that process I really learned how to highlight my skills and how to kind of sell myself to different organizations and really to show them what I had to offer. And that, of course, is an important skill in any application. If you're applying for jobs, you need that. If you're applying for scholarships as a graduate student, you need those. So those have been really helpful as well. I also learned, of course, how to do research, and not only from a technical perspective, but also from a cognitive perspective. I learned how to think about research problems. I learned how to read the literature and look for information in the literature. And I learned how to present. So I had a lot of chances to do presentations on my research while I was at WKU, and those skills were very important as a graduate student, and they're also important even if you don't choose to go into a scientific discipline. That also can be very important in companies. You have to do presentations and things like that. So those were very important skills that I learned. So all of these skills kind of have been really helpful in my post-MHC experiences. So I'm going to go through those a little bit to show you guys where I've been since 2014. So I joined my MD-PhD program in 2014, and for those of you who aren't familiar with how MD-PhD programs work, what you generally do is you do your first 2 years of medical school, which is usually classroom work. So you're in the classroom, you learn about all the different diseases, how they happen, pathophysiology and things like that. And then you stop and you go and do an entire PhD for 4 to 5 years. And then you go back to do the last 2 years of medical school. And in the last 2 years of medical school, you typically are in the hospital and you work on the medical teams and you help the teams take care of patients so that you can get hands-on knowledge and experience in taking care of patients. So by that point, you have completely forgotten everything that you learned in the first 2 years, so it's a little bit of a strange structure, but that's how they do it. So in 2014, I moved to New York City and I started in medical school. And while I was in medical school, I used these kind of long-term planning skills to think forward to my graduate experience that I knew was coming up. And I remembered that I was now going to be eligible for one of the scholarships that I had to decline at the end of my experience at WKU. And I decided to apply for it again. And so that was what's called the National Defense Science and Engineering Graduate Fellowship. So I applied for this, and I used my application writing skills in that. And I also relied on my mentorship network from WKU. So Dr. King wrote me a letter for that, and Dr. Jennings helped me to review the application. So that was very important in helping me be successful in obtaining that grant. So I was able to go into graduate school in 2016, already having a grant in hand. And once I started graduate school, I knew what to look for in a good mentor, and so I was able to pick 2 really great mentors for my PhD research. And those were Carl Nathan and Julian Boberjeks. And so while in lab for the next 4.5 years, I used basically all of these skills. So as I mentioned before, a lot of people in my lab came from different cultural backgrounds, and so it was really helpful to have had that experience communicating with people from different backgrounds. I, of course, already mentioned that I used my knowledge of good mentorship qualities to help me pick good mentors in grad school. Planning and time management, when you're doing research in the lab, it's very unstructured. You have to just figure out what you need to do, when you need to do it, nobody tells you. So you have to be really good at managing your time and figuring out when things need to get done. You have to plan pre-preparations for experiments, and especially in the organism that I was working with, it grows very slowly, so there's a lot of long-term management that has to happen so that you can get experiments done. I also used my application skills, so I applied for another fellowship from the National Institutes of Health later on in my graduate school career, and so I was already very well versed in how to write applications, and so that was a successful application. And of course, the research and presentation skills I've been using all along this path as well. And so I defended my PhD thesis only about 2 weeks ago, so I'm still not used to being called Dr. Schrader. But that was the conclusion of the PhD portion of my MDPhD. So as Margaret mentioned, I will be going back to medical school in August, but before that, I used my application skills again to apply for a critical language scholarship. So I never forgot that I had to decline it in 2014, and I decided that I wanted to apply for it again. So when I did that, I actually talked a little bit with Dr. Grimsley from the Office of Scholar Development, and she helped me kind of structure my application, being that I was coming from a sort of non-traditional background for CLS. She was able to help me figure out how to structure my application so that it would be successful, and ultimately it was successful. So I'm planning on going to South Korea this summer to study Korean. And so after that, I will go back and finish the last 2 years of medical school. And again, I don't remember anything about medicine, so that will be a lot of fun. And after 2 years of trying to remember things and pretend like I know what I'm doing, I will be able to get my MD, hopefully, and I'm planning on doing a residency in clinical pathology. So pathology, the pathologists are the people that you don't see in the hospital, but they're very important. And I know this from some experiences that I had in the hospital before my PhD. Basically, when you have a patient come to the hospital, the doctors don't really know what's up with them until they send samples to the pathologist, and the pathologists tell them what's wrong with the patient. So these guys are the people who are very important in the background. And so in clinical pathology, those are the people who handle the microbiology samples and the people who handle the blood samples and genomics and things like that. So my ultimate goal, I think, is to have a career in microbiology research. I'd potentially like to run a lab in microbiology, and then I can also use my MD to do some clinical microbiology as well. So that's those are my experiences in the Honors College and beyond. And so in the second part of the presentation, I want to talk a little bit about the research that I did for my PhD. So this is sort of a high-level overview because I don't want to go too much into the details. I think it might be kind of not that interesting. So I'm going to try to stay high level. And I want to start out, so what I researched in my PhD was mechanisms of antibiotic resistance in mycobacteria. So I want to talk a little bit about what are mycobacteria, why do we care about them, what is antibiotic resistance, why is it important, and hopefully it will be interesting for you guys. So I want to start out though with why microbiology. So it's always kind of when you are figuring out, as a college student, I know for a lot of people it's hard to figure out really what you want to do and what your passion is. And for me, I was fortunate to figure out that I really liked microbiology when I was an undergraduate student. And some of the reasons why I decided microbiology was what I wanted to do was just because microbes are fascinating. They're very interesting, and these are some points why. They can live anywhere. So these are some extreme environments where you can find microbes, and some of the ones that stand out to me are nuclear contaminated sites, polar, like permafrost, inside volcanoes, in hydrothermal vents, things like that. So they have adapted and figured out ways to live basically anywhere where if you put an animal or a plant, it would die right away. They can eat anything. So some examples of this, I saw this article a couple of years ago and I've remembered it since. Basically, NASA has found bacteria in its so-called clean rooms that aren't supposed to have any bacteria that can survive by eating the cleaning products that are supposed to be killing them. So I find that absolutely amazing that they have adapted to be able to live off something that's supposed to be killing them. And you might have heard some reports in the news about microbes that can degrade oil, that people have thought about using in the cleanup of oil spills. There are microbes that can break down plastic that people are considering using in recycling plastics and things like that. And then I found this article that I thought was interesting a couple days ago. Apparently there are also microbes that they don't really eat them, but they accumulate radioactive isotopes inside their cells. So this is something that people were talking about using to clean up nuclear waste. So microbes are basically amazing, and they can be used as really important tools to do really important jobs in the environment. And basically they're geniuses. So a lot of people don't like kind of personifying microbes because they're different from us in that we can call them geniuses, but they don't really have an intelligence in the way that they do things for a particular purpose. Their intelligence comes from their ability to replicate very quickly and their ability to thus evolve very quickly. But I don't have a problem with personifying them, so I will go ahead and call them geniuses. And this is from an article that was written by a microbiologist called "I Never Met a Microbe That I Didn't Like." I don't think that microbiologist was ever had a very serious infection, or he might probably have a different title for his article. But he had a box in that article called "Microbes Might Be Smarter Than You Think," and some of the highlights from that are they understand military tactics, so they strike quickly with overwhelming numbers, cut the lines of communication, and wear camouflage. They're expert biologists. They've studied biology longer than any other living thing. They've mastered genetic cell biology and immunology. And they always have the last laugh. So they're the first living things we encounter after birth. And when we die, they're the last living cells in our bodies. And then they devour us. And so I think that it's really I really enjoyed this article in all the ways that microbes are much smarter than you think, and much smarter probably than we are. So of course, another reason for studying microbiology is that microbes are very important to human health. And there's both a positive and a negative side to this. So the positive side, you might have heard a lot about the microbiota in recent years. This is a really hot field of research. And so this just involves the study of all the microbes that live in and on us. And they play a very important role in helping to maintain our health. And so that's not what I research, but it is a very important area of research and a very interesting area of research. And of course, the negative side is that microbes can also be pathogenic. They can cause disease in humans and animals, and basically in plants and any other type of organism. So this is more what I study. And so we're going to focus on the pathogenic side here. So we have actually a built-in system to help defend us from microbial pathogens. And so this is our immune system. And this guy here is a macrophage, and he's part of the innate immune system, and this is the part of the immune system that can launch a quick, non-specific attack against basically any kind of microbe that invades the body. So this macrophage here has some other innate immune partners that you can see in this chart, and he also has some friends over here on the adaptive immune system side. And the adaptive immune system is one that takes a little bit longer to activate, but once it's activated, it can launch very specific attacks against specific microbes. And so this is what, if you've been vaccinated against SARS-CoV-2 or against any other kind of pathogen, this is the immune system that's generated. So it has a memory, and it can remember what it's seen before, and if it sees it again, it can launch a very specific and very effective attack against that particular pathogen. So this immune system that we have, it functions pretty well most of the time, and that's why you're not constantly getting infected by bacteria from the environment. And if you think back to the reasons why microbes are smarter than us, the very last thing they devour us after we die, that's because our immune system is not active anymore, so they can invade our body and take advantage of all the lovely nutrients that are there. But sometimes for more serious infections, the immune system can't handle things all on its own. And so for most of human history, there really wasn't anything that we could do about that. So the people who got these serious infections, they just died. But everything changed about 100 years ago when we discovered these compounds called antimicrobials that either slow the growth of or outright kill microbes. And so for bacterial infections, we have antibiotics. For fungal infections, we have antifungals. For a small number of viral infections, we're not really very good at finding compounds that attack viruses. But for some, we have some antivirals. And for parasitic infections, we have antiparasitics. So we sometimes take these types of medications for granted, especially in a country like the United States, but I would argue that antimicrobials are the foundation of modern medicine. So in addition to allowing us to cure infectious diseases, their utility extends well beyond that. They allow us to give chemotherapy to people with cancer because that's immunosuppressive, and so if you have a suppressed immune system, you need antimicrobials to help you stay protected from pathogens. They allow us to do things like stem cell transplants and organ transplants, and then they help keep immunocompromised patients from dying from minor infections. So in short, antimicrobials have completely transformed the way that we practice medicine. But as I mentioned before, microbes are very smart, and they're learning to resist killing by antimicrobials. So antimicrobial resistance has always existed, but it has been steadily and stealthily spreading across both borders and species. So today, many dangerous microbes, such as these ones, which are on the CDC's list of antimicrobial resistance threats, have become resistant to these very important drugs. And because of this, antimicrobial resistance poses a major threat to human health. So here in this chart, you can see the number of deaths per year that are attributed to different causes. And we can see that now, in the current day, AMR takes at least 700,000 lives per year. Now, about 1/3 of these deaths are due to drug-resistant tuberculosis. And this is primarily a lung disease, and it's caused by the bacterial species Mycobacterium tuberculosis, or MTB. So in fact, MTB was the world's most deadly pathogen until it was recently overtaken by SARS-CoV-2, but this is probably only a temporary takeover, and in fact, SARS-CoV-2 is causing probably a resurgence in the number of cases of TB that we will start to see after we've controlled COVID a little bit better. So if nothing is done, the devastating effects of drug-resistant TB and other resistant infections is projected to get much, much worse. So by 2050, which is just 29 years from now, the number of deaths from AMR is projected to swell from the current level of around 700,000 to around 10 million. So this will mean that it will overtake even cancer. And to put that in perspective, if we think about the number of people that have died from COVID-19 in the first year that COVID-19 was traveling around the globe, that was about 2 million. So this number is about 5 times as much. And we've seen the response to this. So hopefully we'll be able to mount a similar response to AMR given the severity of this problem. So these numbers are very sobering, but the good news is that between now and 2050, we have a chance to do some things to try and stop the spread of AMR and try to bring this number down. So there are a lot of things we can do, and among other things, we can practice better antimicrobial stewardship in the clinic, and this means prescribing antibiotics only when they're needed, so for example, for bacterial infections rather than for viral infections. We can limit the use of we can create faster and more accurate diagnostics, so that goes hand in hand with stewardship, and that means that if we can diagnose an infection, that means we can prescribe the correct antibiotic to treat that particular infection. We can limit the use of antimicrobials in agriculture, so for example, in a lot of agriculture, antibiotics are given to animals to boost their growth and not really to cure infections. So we could stop doing that and start giving antibiotics only when animals are sick. And we can also develop treatments that prevent the development of resistance in patients. So this last item here requires a deeper understanding of resistance and how it develops. And so that's really where the work that I did in my PhD comes into play. And this is a little bit detailed, but I want to go into the different types of antibiotic resistance because the types of antibiotic resistance that I looked at in my research were specific types. So there are a number of different ways that bacteria can actually avoid being killed by antibiotics. So to show you these different ways, I'm going to use these 2 diagrams. So the one on the left here is like a microscope snapshot, so you can think of this as showing the bacterial cells that are present at each time point over here on this graph. So this graph is what we call a time kill curve, and it shows us the concentration of viable bacteria over time during the course of exposure to an antibiotic. So here we have 2 rounds of exposure to the same antibiotic, and these are shown in the gray background in both of these charts. And then they're separated by an antibiotic-free outgrowth period where the cells are able to grow without the antibiotic being present. And this is shown with the white background. So this A here is just before the first exposure to the antibiotic. B is after the first exposure. C is after this antibiotic-free outgrowth period. D is after dilution of the cells to prepare for the second exposure. And then E is after the second exposure. So what we're looking at here is what we would expect for a bacterial population that displays no resistance. So basically, when you expose them to the antibiotic, all of the cells die in a relatively quick period of time. So this is what we would want to happen when we treat a patient. We want the antibiotic to be able to kill all of the bacteria so that the patient is cured. But as I mentioned before, these microbes are very smart, and they've developed a number of different ways to avoid this happening. So the first of these is what we call MIC-shifted resistance. This is probably the most well-known manifestation of resistance, and what happens in this type of resistance is that all of the cells are able to grow when the antibiotic is present. So that means the number of bacteria gets higher and the patient gets sicker, even though they're being treated with antibiotics. So in this type of resistance, the MIC, which stands for minimum inhibitory concentration and refers to the lowest concentration of antibiotic that you need to inhibit the growth of the microbes, increases. So this is an assay that we use to look for resistance, and so we can tell that the bacteria are resistant using the assays that we have in the clinic. So the second type of resistance is called tolerance. So in this type, the bacteria can't grow in the presence of the antibiotic, so the number doesn't increase, but the killing rate for the entire population is slowed. So that means that after the end of this first round of exposure to the antibiotic, we still have some cells that are surviving. And so when the patient stops taking the antibiotic here, those cells can grow, and then they can lead to relapse of the infection. So for this population that's grown, if we then dilute it and expose it to the same antibiotic again, the rate of killing remains the same. And then there's a third type of resistance that's called persistence. And so this type is very similar to tolerance, but rather than the entire population, there's only a subpopulation of bacteria, which are called persister cells, and they're shown in orange here, that have a lower rate of killing. So if we look at what that looks like on a population level, we have a biphasic kill curve. So here in the first phase, we have a fast rate of killing, and this is when all these white cells, the susceptible cells, are dying. And then that's followed up by a slower killing phase here. And then this is representative of the killing rate for the persister cells, the orange guys here. So similar to tolerance, this means that after we stop taking this first round of antibiotic, we still have cells that are surviving. And so if the antibiotic is removed, those cells can grow, and they can lead to, relapse of the infection or regrowth of the bacteria. And those guys that are regrown from those persister cells will have persister cells present in the same proportion as before. So if we expose to the antibiotic again, they're going to have the same rate of killing as before, or the same manifestation as before. So what's interesting is that most bacterial populations actually contain a small number of persisters. So if you take a bacterial population and you expose it to antibiotics, you're going to see something like this most of the time. So when we talk about resistance, meaning something that is higher than what we see normally, we usually are referring to something called high persistence. And this is when the proportion of persisters in the population is higher than normal. So that means you have a higher proportion of cells that survive exposure to the antibiotic. So because this tolerance and persistence phenotypes, these don't lead to population level growth, but do lead to increased survival after exposure to an antibiotic, we call them collectively high survival phenotypes. And so the reason that these guys it's important to separate them from this type of resistance is that in the clinical assays that we use, we can only find this kind of resistance because we look for growth. So we might have this kind of resistance present, but we can't tell it's there because we don't look for death of the bacteria in the clinic. We only look for growth. So in terms of where these kinds of resistance come from, there are two major sources. And so the first of these is genetic resistance. And this is when the bacteria get a mutation in their genome or they acquire new genes from surrounding the environment. But there is another source that's a little bit less well known, and this is called phenotypic resistance. So in this type of resistance, the bacteria have the exact same genome as the susceptible cells, but they have triggering of different machinery that is already present in the bacterial cell. And this is usually by stressful conditions in their environment, and that triggers higher levels of resistance in the bacteria. And the reason why it's important to distinguish between these two types is because in a lot of assays that we have in the clinic, we can actually sequence the genomes of bacteria that we take from patients, and we can look for these genetic resistance mutations. But if we have phenotypic resistance, we can't tell that it's present just from sequencing the genomes of the bacteria because it looks like they're susceptible to the antibiotic. So any of these three manifestations of resistance can be achieved through either genetic or phenotypic mechanisms. And usually when we talk about AMR or antimicrobial resistance, we're talking about genetic MIC shifter resistance. So these guys that can grow in the presence of the antibiotic and that have mutations. But no matter the source or the type of resistance, there are important clinical consequences to antibiotic resistance, and that's why we care about it, and that's why we study it and try to figure out how it happens. So for MIC shifter resistance, these include treatment failure with the antibiotics that are normally used to treat a particular antibiotic or a particular infection. And so we have to use alternative drugs to treat that infection. And these drugs usually have disadvantages. They might be less effective, so that means you have to treat the patient for a longer period of time. They might cost a lot more money, or they might have more severe side effects for the patient. So that means that even if the patient is able to be cured, it's usually through a less advantageous method. And in still rare but increasingly common cases, there are no longer any effective antibiotics available. So this is a very scary fact, and this kind of threatens to throw us back into the era before we had antibiotics. So because of these important consequences, this form of resistance is the major force behind the AMR crisis that I talked about a few slides back. But these high survival resistance phenotypes that are types that I talked about also have important clinical consequences. So a standard round of antibiotic treatment might fail to clear the infecting bacteria, and so this can lead to relapse of the infection after the patient stops taking their antibiotic. So to kill these tolerant or resistant bacteria, we have to treat the infection for a longer period of time. And like using alternative drugs for this MIC-shifted resistance type, this raises the cost and also the risk of side effects for the patient. And finally, these tolerant and persistent bacteria, they survive for longer periods of time under antibiotic selection pressure, and sometimes they also have increased mutation rates. So this gives them the opportunity to acquire mutations that lead to MIC-shifted resistance, and then you get all the consequences of those. So we can think of these high survival types of resistance as a fuel for the AMR crisis. So these high survival forms of resistance are particularly relevant to the treatment of tuberculosis, and these are the types of resistance that I studied in my PhD. And we study them in tuberculosis and because of their relevance to tuberculosis. So one of the reasons why they're important for tuberculosis is because MTB is an intracellular pathogen. So it actually lives inside immune cells called macrophages. And inside the macrophages, it's exposed to different stresses that don't actually kill it very well. They're supposed to kill it, but they don't really kill it very well. And instead, they lead to induction of this tolerance and high persistence. So they make the bacteria more resistant to antibiotics. So this is probably the reason why it takes such a long time to treat tuberculosis. So if you've ever had an infection like a strep throat or maybe a skin infection, and you took antibiotics for it, you probably took the antibiotics for maybe 7 days, maybe 10 days, maybe 2 weeks at the most. But for tuberculosis, you have to treat the patients for 6 months. And so this is with a cocktail of 4 different drugs taken for the first 2 months, followed by 2 of those drugs taken for the remaining 4 months. And even after this very long treatment period, we have a treatment success rate of about 85%. So that's pretty good, but it's not perfect. And also this exposure to these antibiotics can also lead to induction of these different types of resistance, so tolerance and high persistence. So that can further complicate treatment. And we've also seen experimentally that these tolerant and persistent cells can acquire mutations that lead to MIC-shifted resistance, and patients can also be infected primarily with cells that already have these resistance mutations. So these drug-resistant MTB is even harder to treat, and recommended treatment regimens are going to vary based on the specific resistance profile of the invading bacteria, but in general, the 4 to 5 drugs taken for a period of at least 24 months, that's almost 2 years, is recommended. And even then, there's only about a 40 to 60% chance of cure, depending on how many drugs the bacteria are resistant to. So for many patients, that means that a cure is not available for their infection, and that means that they die from the infection. And this is one of the reasons why TB is such a deadly disease. So we know a lot about the mechanisms already of MIC-shifted resistance in MTB, but a lot less is known about these mechanisms, the mechanisms of tolerance and persistence. And since these might be important in the evolution of genetic MIC-shifted resistance and also have their own clinical consequences, it's critical that we learn more about them, and that's really what I focused on in my PhD research. So we used a tool in my research that's called something called high survival mutants. So again, high survival refers to these tolerance and high persistence resistance types. And so these types of mutants are ones that have mutations that allow them to be tolerant or to have high persistence upon exposure to antibiotics. And so we can use these mutants as a tool for understanding the mechanisms of both genetic and phenotypic resistance. And these mutants also have clinical relevance themselves, so they have been shown to evolve during antibiotic treatment both in the laboratory and also in patients. So that means that if we know what mutations lead to these types of resistance, we can look for those mutations in isolates of bacteria that are taken from patients, and we can see whether or not they have these mutations that cause these resistance phenotypes, and then we can use that information to design a more effective treatment strategy for the patient. So in my project, basically what we did is we isolated high survival mutants in Mycobacterium smegmatis, and this is a species of mycobacteria that's related to Mycobacterium tuberculosis, but it grows faster, and it also is non-pathogenic, so that means it's much easier to work with in the lab. So we used this to isolate high survival mutants, and then we studied how mutations in genes for the synthesis of the amino acid arginine lead to high survival. So amino acids are the building blocks of proteins, and this particular pathway that we studied is used by mycobacteria to make this building block that it then uses to make proteins. And then we extended our results to the pathogen Mycobacterium tuberculosis, and we showed that similar mutations also led to high survival, or this antibiotic resistance in Mycobacterium tuberculosis. So this here is a summary of the results that we got. So again, if you don't have any resistance, the bacteria will die very quickly upon exposure to an antibiotic. When arginine biosynthesis is normal, it's not mutated, we have a small number of persister cells that survive when we expose the cells to an antibiotic. And in the mutants that I studied, we have arginine biosynthesis that is kind of messed up by these mutations. And so the pathway has a problem. And this leads to upregulation of a gene called YB7, and it's not that important what that gene does, but that was the mechanism behind the resistance phenotypes that we saw. So we saw 3 types of resistance. We saw tolerance and high persistence to 2 different antibiotics. And then we also saw MIC shifter resistance to another antibiotic. And this was all from just 1 mutation. So we think that our results have important implications for the formulation and evaluation of combination antibiotic therapies like the ones that are used for the treatment of TB. So our specific result, again, was that perturbation of arginine biosynthesis in mycobacteria leads to 3 types of resistance to 3 different classes of antibiotics that target pathways other than arginine biosynthesis. So we kind of generalized this result to say that maybe perturbation of any essential pathway targeted by an antibiotic could lead to resistance against other antibiotics. So if you think back to how TB is treated, we treat TB with 4 different drugs at the same time. So possibly, if the TB is exposed to this blue drug here, that might lead to induction of pathways that give you resistance to maybe this yellow drug or this gray drug. And so that will lead to less effective treatment, and that could be some of the reasons behind the long length of treatment that's needed to treat TB. And this could also be leading to the emergence of genetic MIC shifter resistance in some cases. So if we can understand how that happens, then we can develop ways to block that happening, and that could help us to develop faster treatments for TB that work better and that limit the development of genetic MIC shifted resistance. So that is kind of the summary of the work that I've done. So here you can see all the mentors and all the colleagues that have helped me in this project. So I had 2 really great mentors, and I had great people on my thesis committee, and a lot of great people in the lab also contributed to the project. So doing a PhD is really a very collaborative effort between you and your mentors and the other people in your lab as well. And so I'll stop there. And I'll leave a little bit of time for questions if anybody has any. So thank you guys very much. I hope that wasn't too detailed, but thank you.
Dr. Schrader,
Yes.
if you were to give the scholars in the audience 3 pieces of advice on what they should do during their WKU Beginner Honors College time, what 3 pieces of advice would you give them?
Definitely visit the Office of Scholar Development. Make a scholarship plan. Apply for anything and everything that you are interested in or might be interested in. I think that's really important. It helps you develop very important skills. And if you end up winning the scholarships, you also have, that opens up a lot of opportunities for you. And it can really look very good on your resume as well for when you're going on to look for other opportunities, whether that be graduate school or jobs or anything like that. So that's definitely number 1, I think. Number 2, and this is sort of difficult right now, but study abroad was a very important part of my experience. And so if you have the opportunity to do that coming up, I think that is definitely something I would recommend. I really learned a lot in my experiences and gained a lot of important skills. And I think, I guess, I actually had a slide about advice. So I think I already said explore kind of new experiences, new classes, new countries, explore everything. I think college is a really great time to explore a lot of different things. And so definitely do that. Don't be afraid to try out new things. Don't be afraid to change your major if you aren't liking what you're doing. Apply for scholarships, I said. Focus on the positives. So some scholarships you might not get. You might not do well in a certain class. You might have some difficulties with one thing or another. But I think it's always important to try and find the positives of different things. So if, for example, if you apply for a scholarship, you don't get it, focus on what you learned from that process. And I think that can really help you kind of be proud of yourself for putting in the effort, and also help you to really track your development over time. So that was more than 3. But I also have cultivate friendships on here, so I made a lot of great friends in the Honors College. One of my friends even came to my thesis defense because it was over Zoom. And I think it's really important to make, there are a lot of really awesome people among your classmates in the Honors College, and that can be helpful to you in the future from many perspectives. You can help them or use them as a network when you're looking for jobs. You can use them as a network when you just want to talk to someone. So that I think you'll find really great friendships can be made among your colleagues in the Honors College. Find great mentors. I've talked a lot about that. Do what makes you happy. I think that's important, and I think I didn't do enough of that. Sometimes I did a lot of things that made me happy in the Honors College, but sometimes I think in my graduate work I kind of got away from that a little bit, but I think that's very important to always keep that in mind as well.